Now that you've completed the mini PCR Sickle Cell Genetics Lab, it's time to interpret your resulting gel. This video will explain how to use the information in your gel to determine the genotype associated with each sample. Remember that in this lab, PCR was used to copy a 400 base pair section of the beta globin gene. Then, a restriction enzyme that cuts only the typical beta globin allele was added to each sample. The restriction enzyme cuts the 400 base pair PCR product into two fragments, a 150 base pair fragment and a 250 base pair fragment. This means that when you see 150 and 250 base pair bands in a sample, the typical beta globin allele is present. If you see a 400 base pair band, you know the DNA was not cut by the restriction enzyme, and it therefore carries the sickle cell mutation. We'll use the DNA ladder like a molecular ruler to determine the lengths of the DNA fragments in our samples. We'll focus on these bands in the ladder, which contain DNA fragments 100 base pairs, 200 base pairs, 300 base pairs, and 500 base pairs long. The DNA fragments we expect from our experiment fall into this range. In the first lane, we see two bands. This band traveled farther than the 300 base pair band in the ladder, but not as far as the 200 base pair band. We can estimate that it contains DNA fragments about 250 base pairs long. This band traveled further than the 200 base pair band in the ladder, but not as far as the 100 base pair band. We can estimate that it contains DNA fragments about 150 base pairs long. This banding pattern means that this sample contains only the typical beta globin allele, so this sample came from an unaffected individual with the AA genotype. In the second lane, we see one band. The band has traveled farther than the 500 base pair band in the ladder, but not as far as the 300 base pair band. Based on the design of our experiment, we can infer that this band contains the 400 base pair DNA fragments associated with the sickle cell allele. Because this sample contains only the sickle cell allele, we know this individual has the SS genotype that indicates sickle cell disease. Note that while this person does have two copies of the sickle cell allele, all the DNA fragments are the same length, so there is only a single band on the gel. In the third lane, we see three bands. Comparing them to our ladder, we see that this band contains DNA fragments about 400 base pairs long, this band contains DNA fragments about 250 base pairs long, and this band contains DNA fragments about 150 base pairs long. The 400 base pair band represents DNA from the sickle cell allele, and the 150 and 250 base pair bands represent DNA from the typical beta globin allele. Therefore, this individual has the AS genotype indicative of the sickle cell trait. Now you can compare the Patel family samples to these results to identify the genotype of each family member.